So the reasons that we breathe should have become evident to you guys in the last chapter when we were talking about cellular respiration as a component of metabolism. That the way to efficiently get energy requires oxygen. And oxygen only comes in really through your alveoli in your lungs. And also important to this process, of course, is getting rid of carbon dioxide. And that is why we call this the respiratory system, because respiration requires an exchange. So, of course, when you did cellular respiration, right, oxygen had to come in, and it's kind of cut off right here on the screen, right here, and carbon dioxide was given off. So the oxygen is used, and used constantly, and at different rates in the cells of your body, depending on how much energy they need. And for every oxygen used, of course, carbon dioxide is produced. And so cells constantly have a high level of carbon dioxide and a low level of oxygen, which helps to drive the continued movement of these two gases between the cells of the body, the blood, and the lungs. So we want to look at, we got the why we breathe. We want to see how we move these gases, because we are very big multicellular organisms, so we need some pretty sophisticated tubes to pull this air in and push this air out. So I say the system consists of many passages. So most of everything that you can see with your eyes that you consider the respiratory system, right, trachea and bronchi and parts of the oral cavity and nasal cavity, these are all just sophisticated spaces and tubes to move air. They're not going to participate in this exchange that we get the name for. All right, the exchange is going to go on at a very, very small level, microscopic level. And we have to remember that, like all the other systems of tubes we've looked at, the histology of the tube has to change to allow this process to occur. And we want to know why. All right? So, again, here with the why we breathe, cellular respiration, oxygen needed, ATP produced, carbon dioxide removed. Right? So, to get to this point, right, on the cellular level, we have several steps that we're going to look at in this chapter. First and foremost, we have ventilation. Ventilation, like if we talk about your room ventilation, it's just moving the air. All right. Next, we have the actual exchange. So this is going to occur with all these microscopic structures down within the lungs themselves. So down to what we call respiratory bronchioles and alveolar ducts and alveolar sacs. Then this oxygen moves into the blood and the pulmonary capillaries, and it moves out, right? Carbon dioxide moves out of the pulmonary capillaries. And so we look at, in this chapter, how we transport the gases, because these gases don't stay bound. They don't, you're a liquid environment, so we have to figure out ways to keep a gas in a liquid state for delivery. And then the delivery is really considered the internal respiration. So that's the exchange of the oxygen, so oxygen out of the blood and into the cells, and carbon dioxide out of the cells, right, and back into the blood everywhere in your body. All right, so here's an overview of what we talk about as the anatomy, right? And again, everything you can really see is dealing with ventilation. So I say, provide oxygen. Here's what our functions are. Provide oxygen, remove carbon dioxide, maintain acid-base balance. Oh, wow, I didn't mention that in the last chapter, in the last slide. But maintain acid-base balance, okay? Well, when we produce carbon dioxide combines with water, and of course this is all part of the metabolic process, right? We call cell respiration. Carbon dioxide and water are produced as these nutrients are broken apart. When carbon dioxide and water get together, they make carbonic acid. And so every time that you produce carbon dioxide, you're producing acids. So we have to be very careful. So the more carbon dioxide produced, the more acidic you become. So the respiratory system, in expelling carbon dioxide, is also helping to balance your pH in your body. All right? So the two divisions, as I've alluded to here, are conducting, and this is going to deal with ventilation. So moving the air in and down to the alveoli, and moving air out. All right. So And so the conducting division also removes debris and pathogens, right? So we have modifications to these tubes which help us, including lots and lots of mucus and macrophages. It has to warm and humidify air. So again, air out here that we're pulling into the body has to be at the right conditions. So that's up to the conducting division. And you learned in AMP1 that you also get a sense of smell, which is a very important sensory for survival through this part of the respiratory system. Most of the tissues actually, and all of it, again, none of the respiratory can be seen. So most of this tissue is going to be, and most of the tissue of the respiratory system is in the respiratory division. All right, so there's an awful lot of very, very, very small structures in the lungs, about 300 million alveoli. And so you really have to consider that, all the stuff you can't see is the major part and does the most important aspect of the respiratory system. And that's where we get exchange. So, as I said before, this division will consist of respiratory bronchioles, alveolar ducts, and alveoli. And you have one model in lab that kind of more or less shows that. Most of the models in lab are showing you, of course, the gross anatomy. All right. So moving through each part here to get to the final, get to the point where we can actually exchange. We start with the nose. There's lots of aspects about the nose to know, thrill your friends with, but I'm not going to ask you these terms on a test. Of course, most of the nose is hyaline cartilage, and there's just two little bones, that appropriately called the nasal bones, that support this, and otherwise, like I said, a lot of cartilage. All right, so I say here lots of neat anatomical terms you don't need to know unless you're a facial reconstructor. The bone is nasal here, as I've said, connected to the maxillary bone, right, this upper jaw. The nares are these openings here, open to the nasal cavity. And the walls are made up of the ethmoid bone, right? It has two conchae in the ethmoids, and you know those for lab, right? And there'll be a superior and a medial concave. And the third is actually its own bone called the inferior nasal conchae. All right. You have a number of sagittal heads in lab, and so this should be very similar and very familiar to you guys at this point. They generally end about here, right as the esophagus begins, so do bear that in mind. All right. So here's those conchae, which are basically shells in the nasal cavity. All right. And they're going to increase surface area in this space we call the nasal cavity. And that's really important, again, for this process of warming and humidifying air. All right. They project in there, so they actually disrupt flow. As air comes in through the nares, all right, you get this, they hit these shelves, and this creates turbulence. And so air is stalled here for a short period of time, which is good. This helps clean it up, too, because you have all kinds of this mucus that's being secreted in here, and then cilia that move this. And it helps conserve water when you exhale. So it's not just moving right out, because remember, there's water in, as carbon dioxide leaves the body, it leaves with water, and we want to recapture that water. So this allows for that to occur as well. The floor, of course, of the nasal cavity is the hard palate made up of the maxillary bone and the palatine bone. Sinuses are scattered. They're called the paranasal sinuses. You have a frontal, you have a sphenoidal, you have ethmoid, and you have maxillary sinuses. And they open to the nasal cavity, and they also help to warm and humidify air. So sinuses produce mucus, right? They produce excessive mucus and fluids. You get sinus pressure, and these sinuses help to lighten the weight of the skull. All right? This is a lovely picture of the histology of the nasal cavity, and this is maintained really throughout most of the gross anatomy of the what we call the conducting division. Right? This is basically pseudostratified and ciliated, right? Cilia to move all that mucus, because if mucus stays stagnant, it is actually a lovely sugary protein that bacteria love. So that's why when you're producing a lot of mucus from an allergy, it's very easy to get a sinus infection, right, or a bacterial infection, because all that lovely, yummy mucus can be a source of food for the bacterium. So the cilia, again, move this, get it out of the way, right? The capillaries, and there's tons of these little capillaries in this submucosal area here that warm, right? There's glandular cells that secrete lysozyme. So remember that's the enzyme that breaks down, disrupts the cell wall of bacteria. So you've got some chemical defenses besides the mucus that is really a barrier defense. These cells produce defensins, and then you have patrolling immune cells here too. Pretty convenient. Remember, defensins, they punched holes in the membranes of cells. All right, so the pharynx, so air passes on through the nasal cavity and into the pharynx, which is nicely color-coded here for the 3 divisions that you have to know for lab as well. So you've got the nasopharynx that will be from the rear of the nasal cavity to the uvula, the last part of the soft palate, and then the oropharynx from the uvula to the epiglottis, and then the laryngopharynx from the epiglottis on down to the thyroid cartilage that you see right here. So the pharynx is a muscular tube, right? It's just there to move, just like we talked about in the digestive system, all right? With 3 regions that let air pass here, and then, of course, down here, liquids and food. So the pharyngeal tonsils are right in this area, as you remember maybe from your models, but you can see them on your models. And this is the opening to the eustachian tubes, which was continuous with the middle ear. The oropharynx allows air and food to pass, and so this pseudostratified columnar, ciliated columnar epithelium does become stratified squamous in this region, all right? But that's not a big deal. It's going to return to pseudostratified again when we get into the trachea. You have the palatine, right, which are right about here, and then the lingual tonsils, which are, of course, the posterior aspect of the tongue to protect us here. I said the laryngopharynx then diverges into the larynx and the esophagus, so we have a choice to make, all right? Now, most of the time, the epiglottis, because most of the time you're breathing, the epiglottis is open and transitioning, right, from the laryngopharynx into the trachea, through the larynx into the trachea, because that's all larynx right here. And when you swallow, of course, this rises up, and the larynx rises up, the epiglottis comes down, uvula goes up, and we really direct food down the esophagus. All right? Excuse me. We have plenty of larynx models in the lab, and we do ask you to know there are 9 pieces of cartilage, 3 of which are paired. So 6 of them have 6 pieces or pairs, and then there's 3 singular. So it's mostly hyaline cartilage with the exception of the epiglottis, which is elastic cartilage because it's going to be stretchy, right? And it's going to undergo a lot of stretch and recoil. So cartilaginous structure connects the laryngopharynx to the trachea, all right? Regulates the volume of air entering and keeps non-gases out. So, right, the idea is no liquid or solids should go down the trachea. All right, and so, and this epiglottis is a huge protector of that, right? And again, regulates volume because when we look down in, okay, these are sagittal views and this is a coronal view, but if we looked down in to the larynx, you'd see this opening, right, that is created by the vocal folds and the true vocal cords. And these really make up this opening called the glottis, and the glottis allows, and you can change the diameter of that glottis to control air entering and leaving. And that's very convenient for a lot of different things. So we have, again, a lot of features on here to show you this idea. There's lots of membranes to hold this together. Here's the hyoid bone and the epiglottis. The cuneiform, you're going to have to learn, and that's paired. The corniculate sits on top of the arytenoid. All right, so this is kind of giving you this idea. So these 3 are paired. The epiglottis is by itself a singular. The thyroid cartilage is anterior big shield, and the cricoid or cricoid is an actual ring. All right? So again, 3 large unpaired see from the anterior view, right? The epiglottis, the thyroid, and the cricoid. And then 3 small pairs attached to the epiglottis and vocal cords. So that's what you see here. Right? We transition from the stratified squamous of the pharynx to pseudostratified with cilia again. And again, the epiglottis epi on top of the glottis will help to close off that glottis. All right, so here's looking down into the glottis I was talking about. So the glottis is made up by this opening, right? And then these, right, true vocal cords, which are made of again, an elastic, regular connective tissue. And then these vestibular folds, which are more mucus and related soft tissues, and sometimes called the false vocal cords. And so here's again our tongue, so here anterior. The glottis is collectively all of this. You've got a vestibular fold, you've got true vocal cords,